Hi, so today I'll be talking about the structural insight into maternal embryonic Leucine kinase, conformation and inhibition toward structure-based drug design. Maternal embryonic Leucine kinase was mentioned as MELK. I would be referring to MELK because of its short acronym. Before I talk about kinases, before I talk about MELK, I would like to give a brief introductions on the kinases. Kinases are enzymes that catalyze phosphorylations. Phosphorylation is a process of transferring a phosphate group from high energy molecules as ATP and putting it on a substrate, and the byproduct would be ADP and the phosphorylated substrate. Kinases are important in cell metabolism, cell regulations, and protein regulations. They also contribute to many other cellular pathways such as glycolysis. MELK are the member of AMPK family of serine-threonine kinases. They are unique in that they get activated by autophosphorylations in the T loop, specifically at threonine 167 and serine 171. They are important in cell cycle regulation, stem cell renewal, and progenitor cells renewal, and also they regulate apoptotic functions. Apoptosis is the process of degradation of unnecessary or cells that are potentially harmful to our bodies, such as tumor cells. And MELK phosphorylates a broad spectrum of substrates that would then do the function. If we take a look at the MELK structure, we can see that MELK comes with this EPA domain, and this is unique in MELK, only MELK kinase. The other kinases don't have this EPA domain. EPA is an ubiquitin-associated domain. The function of ubiquitin itself is it degrades superfluous or defective protein. But if UPA domain, the function of UPA domain is still undetermined, and it only shows on this MELK kinase. Now, the structure of MELK kinase is mostly in two parts: N-terminal, C-terminal. N-terminal is composed of mostly beta sheets. There are 5 beta plated sheets, and there is only one alpha helix, which is known as alpha C here. And C-terminal is almost entirely composed of alpha helices. Now, the UPA domain is bound to N lobe, and it is connected with this long 20 amino acids unstructured linkage to the C-terminal. Also, here is the active site right here, and this is the hinge region right here, and this is the opening of active site. Now, what is worrying about MELK if it is important to cell cycle regulation? MELK has been found overexpressed in various undifferentiated tumors such as breast, prostate, and brain. It has been also found that they play a significant role in breast cancer cell growth by phosphorylating this protein right here, BC1GL. Now, this protein promotes pro-apoptotic function, but when MELK phosphorylates this protein, it inhibits its activity and thus promote cancer cell growth. And also, overexpression of MELK correlates with poor prognosis of patients with glioblastoma, breast, and prostate cancer. What does that mean is when MELK is overexpressed, the symptoms in those patients with these cancers are hard to track. So what could be done to MELK? Now, knocking MELK completely over is not a good idea, as it is important in cell cycle regulation, but inhibitors could be introduced to suppress its activity in tumor-related cells. So what are the inhibitors out there? There's AMP-PNP, which is ATP analog, a competitive inhibitor of ATP. Now, the difference between ATP and AMP-PNP is there's an amide group in AMP-PNP, which stands for adenylyl imidodiphosphate. Now, other two inhibitors are benzodiazepyrazole and pyrrolopyrazole, also known as CPD1 and CPD2. So what are the groundworks and hypotheses of this research paper? They have done high throughput screening of their proprietary compounds that are kinase targeted. And they have done this high throughput screening using a kinase glow assay. Kinase glow assay used in 384 well plates, where each well will contain kinase, the enzyme, ATP, and a substrate that the enzyme will phosphorylate. Now, the reactions was measured without adding any inhibitors first, and after the end of the reactions, the leftover ATP was measured. And in the second reaction, the inhibitors were added to the wells at a different concentration, and the leftover ATP was measured again, and from the two, the IC50 value of its inhibitor compounds was determined. IC50 value stands for inhibition concentration by 50%. Now, they have found good activity of CPD1 and CPD2 on MELK. They then did crystallographic structure analysis to characterize the active site of MELK kinase, and their hypothesis provide detailed structural insights into the protein and inhibitor interactions in a view of structure-based drug design approach. Now, they have selected these two CPD1, CPD2 inhibitors, and they have experimented these two inhibitors with 50 other kinases. What we see in this figure is the first inhibitor inhibits MELK activity by 50% at a concentration as low as 0.027 micromolar, and it also inhibits 3 other kinases of the 50 that were tested at a concentrations lower than 1 micromolar, whereas CPD2 does it at 0.012 micromolar to inhibit MELK activity by 50%, and it also inhibits 7 other kinases activity by 50% at a concentrations lower than 1 micromolar. Now here what we see right there is that the concentrations of CPD1 requires more than 10 micromolar on this many kinases among the 50, whereas CPD2 requires 10 micromolar concentrations less than what the amount of kinases that were required for CPD1. Now here my critique is I would have liked to see this similar experiment on using this AMP-PNP inhibitor, as it is the main competitive inhibitor and an analog of ATP. If they had done it, then it would have been better, or we could have seen whether these concentrations right here, whether they are really small or really big. We could have compared them well. So they have performed crystallographic structures of MELK with or without the AVA domain. What they have shown in the paper is that without the UPA domain, there was less yield of purified protein, which was unsuitable to form crystallographic studies. Whereas with UPA domain, high yield of purified protein was formed, and there is stable crystallographic structures also was available. Now we see that UPA domain has 3 alpha helices. Alpha 1 is anti-parallel to alpha 3 when it's bound to the MELK complex and joined by alpha 2. When it's not bound, it has this N-canonical shape, and alpha 1 and alpha 3 are parallel instead of anti-parallel. And here's the structure if it was this U shape bound to MELK with a lot of interactions with the alpha 3 and N lobe of MELK. But if it was on the N shape, there would have been less interactions on the alpha 3 helix with that N lobe. Now, if we take a look at the residual level interactions, we see there's a lot of hydrophobic interactions going on with the alpha 3 helix and with the N lobe of MELK kinase. Now, interestingly, we should look at the polar ends of alpha 3 helix. We see there's a histidine 312. Now here's the side chain of histidine with this imidazole group right here, and the backbones is right here. There's this amide group. The blue represents amide or amino group, and the red represents the carbonyl or carboxyl group. So, and this histidine 312 is mediated interactions by water molecule with tyrosine. Again, this is the side chain of tyrosine, backbone of tyrosine, the carbonyl group of tyrosine, and leucine, amide group of leucine behind this beta pleated sheet. And on the other end, lysine 323 is bound. There's the side chain of lysine 323 and is bound to glutamate 35. Here's the side chain of glutamate, the carboxamide group of glutamate. This is the side chain. And threonine right here, this is the side chain, but this is the carbonyl backbone. So lysine 323 is bound to the side chain of glutamate 35 and backbone of carbonyl threonine 33. Another important interactions to look at is tyrosine 310, which resides between alpha 3 and alpha 2 of the UBA domain, and it forms stacking interactions with the N lobe of MELK kinase. Here is the lone alpha C helix lysine 62 residue, and there's another residue coming from this beta pleated sheet, which is leucine 72. They form stacking interactions with tyrosine 310, which is very important, keeping in this alpha helix on the right positions, which help the active site stabilize. MELK in complex with the first, the ATP analog inhibitor AMP-PNP was resolved at 2.6 angstrom, and we see the hydropolar interactions of the inhibitors and the active site. What we see here, the adenine group, there's a ribose group, and there's the phosphate group, and here's the amide of the inhibitor. Now, adenine group, here is the nitrogen group. The nitrogen atoms of the adenine group is bound to the backbone carbonyl of glutamate 87 and backbone amide of cysteine 89. Here is the cysteine. Now, the ribose moiety is forming interactions with the glutamate side chain. This glutamate 93 is coming from the C terminal. And the first two phosphate are forming interactions with lysine 40, which is being held by glutamate 57 side chain. And here's this amide group, and another phosphate oxygen is bound to aspartate 150. Now, what we see here is there's the inhibitor, aldose inhibitor, it has similar interactions as it was with the ATP with this active site. Now, MELK in complex with CPT1 was resolved at 2.2 angstrom and shows similar polar interactions as it was with AMP-PNP. First of all, you look at, on the left side, hydrophobic interactions of CPT1. We see a lot of hydrophobic residues which forms hydrophobic interactions. And the polar interactions, on the other hand, the benzodiazepyrazole core is right here, is forming interactions with, again, the same residues, GLU87 backbone carbonyl and cysteine amide backbone with the nitrogen group. It also has the nitrogen groups. And there is what is additional to see here is that the lysine, amino group of lysine, and the carbonyl here is bound via a water molecule, red dot representing water molecule. And the amide group right here is mediating interactions by another water molecule with glutamate 93 coming from, again, the C-terminal lobe and the glutamate 136 backbone carbonyl. And here, this nitrogen of this propolic amino group is bound to aspartate 150 side chain and side chain of asparagine. Now, again, as you can see, we see that there is similar interactions going on with the active site and the inhibitors, again, similar to AMP-PNP and the active site. But interestingly, this meta-fluorophenyl ring group, the presence of this shifted the tyrosine group toward the solvent. So there is a big shift of tyrosine of the active site, which you don't see with either AMP-PNP or in the next with CPT2. Again, CPT2 in complex with MELK was resolved at 1.83 angstrom, and here is the hydrophobic interactions. Interestingly, there is an aromatic ring here and cyclopropyl ring here, which forming additional hydrophobic interactions, especially this aromatic ring form a bulge, again a bulge in propolyl group as well, cyclopropyl group as well, but this aromatic ring is forming interactions with the alkylic group of the lysine 40. Polar interactions similar, pyrolyl-pyrazole core is right here, forming again interactions with GLU87 and cysteine 89 backbone, amide and carbonyl, hydrogen donor, acceptor, donor linkage, and there is also water molecule mediating interactions, lysine amino group of lysine 40 and carbonyl group of the inhibitor. Another water molecule mediating interactions again with this same residue, glutamate 93 side chain and carbonyl and amide group. And there is the last pyrazole is forming interactions with the aspartate and asparagine via a third water molecule. Now, if we compare the CPT1 and CPT2 inhibitors structures with MELK, we see that this tyrosine residue in blue, this is the position where it would be shifted when CPT1, the first inhibitor, was present because of this meta position fluorophenyl ring. But in the white position is the actual position of tyrosine 88, but it is shifted when this CPT1 is the first inhibitor would bind. But if the second inhibitor binds in this position, there would be no shift. Again, in the active site, also, if we look at that interface area, we see that the interface area of when CPD1 is bound is about 390 angstrom square, which is smaller than when CPD2 binds. CPD2 has interface area of 511, which is like 120 angstrom square more than CPD1. Another interesting factor here is this bulge is coming from that aromatic ring, and CPD2 also plunging 1 angstrom deeper in the active site than CPD1. But my question, so take here, is what about the cyclopropyl group that they have shown right here, which is also forming a little bulge? They haven't mentioned anything about the cyclopropyl group. I'd have liked to see, or would have liked to, at least would have liked to see it was mentioned on the paper. In conclusion, what we can say is that complete crystal structures of MELK with UVA and with the inhibitors were performed, and it was determined that CPD2 is a better inhibitor than CPD1. So their work definitely characterized the active site of MELK and how the inhibitors and the kinase interact with each other. This work would definitely lay the basis to find better, with higher potency, inhibitors that could potentially bind in the active site and suppress MELK overexpression, and this can reduce tumor cell growth as a drug design approach. For future studies, what could be done? They could look for biomarkers that are expressed when MELK are overexpressed. Also, organometallic cofactor calcium, which is found to be MELK inactivators, or reducing agents DGT, which are also found as MELK inhibitors. This could additionally be used along with their inhibitors to see overall they play any role together or inhibit maybe better MELK activity. So that is it. Thank you.